Okay, hello everyone. I hope I'm online. Let me check who has joined us. So Rajdeep, Hiten, Pritam, welcome everyone. So today's discussion topic is about innate immune system. I hope my voice is clear. Is it clear, guys? Is my voice clear? Let me just cross check in my phone. Is my voice clear? Yeah, it's clear. So great. So I'll wait for you guys to join. But एक चीज़ मुझे बताओ, लास्ट दिन का लेक्चर कैसा लगा? समझ में आया ना? It was handwritten, but was it not understandable or how was it? Like, give me some feedback in the live chat such that I know. So hi Rashika, hi Saloni, Pankaj, hi to everyone. So today we would discuss the concepts of innate immunity. Obviously we would draw parallels and some stories would be always there. But guys, also what we need to do today is to solve some questions. Okay, that was important. So today's question solving would be from last day's topic, and let's see who can score them, like who can score the best, right? And these questions would be exactly the type that could be asked in exams, not too much hi-fi type. Okay, so hi Saloni, hi Soumya, hi Shipra, right? The boards would be available soon, so I'll give the boards. Some edits need to be done. I saw some problems, some cases the handwriting is not understandable, so the boards would be available very soon. Okay, don't worry. Oh yeah, here there is no option for poll, so you just need to answer in the comment. Okay, so no problem, you just need to answer in the comment. Okay, great guys, so shall we start? Okay, so before we start innate immune system, let me ask you guys, what do you really understand when it talks about innate immune system? Like, what comes in your mind when you hear the term innate immune system or innate immunity for say? So something should come to your mind, and each time tell me that what comes in your mind, such that I would understand that how much depth I need to cover or where I have to start. Okay, so innate immunity, like the front line, whereas the strong immunity is the adaptive immune system, right? But they are in the secured line and they need some time. So tell me guys, like if innate immune system is so much ready, it has fast response kinetics, why do we need an adaptive immune system? Have you ever wondered? Why do we at all need an adaptive immune system? So innate immunity से तो सब हो जाता, तो adaptive immunity का ज़रूरत क्या था? So guys who are online, please say hi, such that I know who at least who are online. Okay. So any idea guys? So here is a concept that is kind of deeply rooted, but I would try to change it. So they are saying that innate immune system are non-specific. I would like to use the term limited specificity rather than non-specific. And in a moment, it would be clear that why I am stressing on this term, right? So some cases, Soumya and Saloni, you are right, partially right. Some cases innate immune system is not so strong and many pathogens can devise ways to like evade the innate immune system. In that case, you need a fail-safe mechanism such that, hi Gitanjali, so in that case you need a fail-safe mechanism by which you can ensure that your body does not run into the risk of problems, right? So already there is a backup mechanism, and adaptive immune system is a fail-safe mechanism kind of thing. But don't worry, adaptive immune system can also fail, so there could be failure at any time. So it's not a perfect system, but it's a good system. Okay. Yeah, so this is very important point that Monica said. So memories, innate immune system don't have memory. What is memory? We'll talk about it. So let us begin to understand, right? So the first level of like innate immune system is basically physical barriers, such as our skin, right? Such as our nasal epithelia, such as our like gastric juice, etc. So that have, or let's say a lysozyme present in your saliva. These things are basically physical barrier, right? Now physical barrier is just like a fence. So already you have a fence, so the pathogen would at least face some trouble for getting in. It's not so easy for them to get in, right? So hi Devika. Okay, hi Devika, welcome to the stream. I hope this would be very useful. But stream चलते चलते बता दो आप लोग, प्लीज शेयर कर दो अपना ग्रुप में, अपने अपने कॉलेज के ग्रुप में, अपने अपने व्हाट्सएप ग्रुप में, such that other people can also join us and let it be a very big stream, a very big live one. Okay, so the skin is a barrier. Why? Because it can, prevent direct entry of pathogens, right? Now our skin has specific molecules which mediate these kind of counter pathogen kind of like a response. These are small peptides generally, and they are known as antimicrobial peptides. Okay, they are known as antimicrobial peptides. So they have properties which can kill the microbes in various ways. So some of them are actually S100, lysozyme, cathelicidin, defensin, lactoferrin, etc. So these are some proteins which are already present sometimes in our secretions, sometimes in our skin, etc. Okay, such as also psoriasin, etc. So for example, this was discovered. So do you know the story, or can you imagine how this was discovered, that our skin work like a physical barrier? It was a fun story. If you guys want to know, I can tell you. Otherwise, we can just go to the conventional way. So, do you guys want to know this thing? The story behind discovery of. Okay, so one day, one scientist was working on bacteria. Okay, so one day, what happened? So you know, in bacterial hood, you need sterile techniques to culture the bacteria, and one day by mistake he placed his hand on a plate, bacterial plate, on the media. Okay, so he forgot to throw that plate. Okay, right? He definitely sort of put some E. coli there, some plating he had done, but when he was trying to put the lid back, he accidentally touched this plate, right? And eventually he thought there would be contamination, so no risk, he should have thrown that, but he didn't. Next day, what he observed, that the plate which he touched has a finger impression on the plate. He was surprised that what happened, why the places where I have touched is devoid of bacterial colonies. So he could not understand. Later on, next day, what he did, so he took a bacterial colony plate and then he really washed his hand with just some water, lukewarm water, and then put that watery substance into the plate. Next day he counted the number of colonies between one control plate and one plate which has the water and everything. Found out the number of colonies were very less. That means there is some kind of soluble substance present in our skin or hands that can really prevent this pathogen attack. And later on, with advanced biochemical techniques and extracrystallographic techniques, they figure out the nature of these peptides. Then first one was psoriasin, then defensin, cathelicidin. These things were actually discovered. Okay, so we just need to know that there are antimicrobial peptides all over our skin. Otherwise, imagine like why bacteria don't grow on our skin. Our body temperature is just appropriate for them to grow. They are generally sort of, I don't know about how calcarine was discovered. I don't know actually about this. I'll take a look, okay? But imagine like we are exposed to pathogen everywhere. For example, we wash our hands with soap and everything. Let's say before eating, we just wash our hands in the tap water, right? And in this case, tap water also have bacteria. Just for example, if you have access to any microbiology lab or anything, you can just take a little bit tap water and put it on a plate. Next day, sure shot you are going to get some growth. Okay, so this is very important. So our skin always protects us from these kind of pathogenic attack, right? So let us move on. So let us look at some kind of like players of the innate immune system. Okay, so two major players of innate immune system are antigen presenting cells, which are actually macrophages and dendritic cell. Now B cells are also antigen presenting cells, but they are not exactly innate immune system components. Okay, so they are considered as adaptive immune system components. We'll talk about that later, but right now let us focus on macrophages and dendritic cell, because they are the stars for innate immune response. Okay, so you are already familiar with the macrophage and dendritic cell. I would also remind you that where they are born. Dendritic cell, we found that they were a little bit unusual, right? They are not born from either myeloid or lymphoid. They could be derived from both of that, right? So they were a little bit unusual in terms of development. Whereas macrophage is just the differentiated form of monocyte. Monocyte was derived from lymphoid or myeloid. Which one? Okay, but both these cell types are actually antigen presenting cells. That means they can present the antigen to T cells with the help of class 2 MHC molecule. Anything which uses a class 2 MHC molecule, that is known as basically classical antigen presenting cells. So very good, it's myeloid who is giving rise to monocytes. So basically the antigens are presented to T cells with the help of class 2 MHC molecule. And next of that, so these cells have toll-like receptors. So we already told what is toll-like receptor. These are pattern recognition receptor. I'll tell you a story about toll-like receptor, how it was discovered. It's another fun story. So stay tuned, because if you don't stay tuned, you would miss that. Okay, so it's very interesting story, guys. Okay, now overall macrophage and dendritic cell, the way they counter the pathogen is basically they engulf the pathogen. They're highly phagocytotic. Okay, that's the important part. So virus, bacteria, whatever they found, they sort of engulf it. Okay, they phagocytose it. So is it clear, guys? So you understand, right? Now let us focus on these cell type one by one. Let's try to understand how they recognize, how they are developed. A little bit we'll recapitulate whatever we have learned. So yeah, it's a quick recap. Then we are going to look at their function. Then we are going to look at their specificity of they are specific or they are not specific, or they are limitedly specific. So these aspects we would look at, and then we would move to dendritic cell. Okay, so first of all we talk about macrophages. We know macrophage are derived from the myeloid progenitor cells, right? The myeloid progenitor cell give rise to monocytes. Now monocytes are secreted into the blood. Now once monocytes are secreted into the blood, they are in the circulation, but eventually they leave the circulation, leave the blood vessel by cytoskeletal rearrangement. So we'll look into this process in bit more details later on, that how a blood cell can really squeeze through these narrow blood vessels. It's an interesting mechanism and known as diapedesis. We'll talk about it later, but let's just focus on monocyte. So this is how kind of a monocyte would look like while this monocyte is in circulation. But when this monocyte would enter the tissue space, it would be differentiated into macrophages. Now looking at macrophages, understand that macrophages have like finger-like projections, they have like cytotoxic granules, they have more granules, okay, more endosomes, etc. They have definitely more mitochondria. So one great feature about macrophages and monocytes, that macrophages have more mitochondria than these monocytes. Okay, and I'll tell you why these mitochondria would be very important for macrophage rather than these monocytes. It's a very interesting story, right? Great. So yep, let's talk about. Now we have macrophages, right? Macrophages are actually patrolling police officer. We kind of compared them with a patrolling police officer. So generally these police car would patrol all around the city looking for crimes or anything. So generally macrophage does that. Macrophage and dendritic cell, they are pretty much motile. Generally macrophage don't leave our tissue, but for example in the skin, the macrophages are known as Langerhans cells. So they reside there. They also sample these kind of potential pathogens, right? And they're just closer to your physical barrier. In this case it's a fence, but you can understand for our body it would be just beneath the skin, right? Okay, so let us think about a pathogen invasion. Let's say a bacteria has invaded. That is like a terrorist or some kind of agitator has came in through this barrier. They have broken this barrier. But question is, how does our body knows that there is an invasion? So body need to have mechanisms to understand this, right? Yep, exactly. So whenever they come in, they would cause problems, they would cause turmoil, right? So they would start firing, let's say, and there would be bullet shells, empty magazines, etc. That can be actually recognized, these signatures can be recognized by our patrolling police officer, and thereby they would understand something has gone wrong. They would try to encounter, right? So the same scenario, let us try to understand here in a context of our body. So everybody is with me, so please in between say that you are with me, or do some gestures. Please ask questions such that it also motivates me, otherwise it would be very monotonous, right? So you guys are with me? Yep. Okay, so right now we are looking at a tissue space. Maybe it's a hand, right? So it's your hand, and you accidentally prick your hand with a nail. In this case it's a rusty nail on a bench, let's say, generally in your college bench. So with this rusty nail, there would be many pathogens, maybe bacteria, maybe virus that can come in, right? So immediately you know, just beneath your skin there are resident macrophages, there are dendritic cells, which would quickly come near these pathogens, right? Right, guys? So first they would engulf them, but before they engulf them, they need to understand two things. Like, they would first try to kill it, but also would try to know or gain intels about what type of pathogen that was. Was it a virus? Was it a bacteria? Was it a helminth? Was it, let's say, a parasite? These kind of information they would try to know. Question is, how do they know? So biggest question is, how does the macrophage or DC knows the nature of the pathogen? Okay, so how do they know, right? So that's a very interesting topic. So let us try to understand how do they know. So guys, these macrophages have specific receptors on them, which are known as pattern recognition receptor. कल से बोलते जा रहा हूँ, pattern recognition receptors. One popular pattern recognition receptor is basically toll-like receptor. Okay, so PAMs are different. So you just sort of evoked a term known as PAMs. Okay, so what are PAMs? So PAM stands for pathogen associated molecular patterns. Okay, so that means PAMs means it's a part of the bacteria or virus, maybe exotoxin secreted by the bacteria, maybe a component of its cell membrane, cell wall, or flagella, etc. Right? For example, I'll say the flagella of a bacteria. I'll draw it for you guys. So let's say this is a bacteria which is a potential pathogen for our body. Okay, so this is the flagella, right? So this flagella is made up of protein flagellin. So this flagellin is actually recognized by TLR4. So TLR4 kind of recognize bacterial flagellin and alerts these macrophages, on let the macrophages know that this was a bacteria kind of infection. So the question is, what these bacteria would do, what these macrophages would do after they recognize that it's a bacteria or virus, right? So recognition is important, but what is their plan of action? That is our topic of discussion. So let's try to understand that how these cells or these PAMs actually, how these PAMs are actually recognized by TLRs And what does these macrophages do? So these macrophages uptake a hell lot of oxygen, and this is known as oxidative burst. Now with the help of an enzyme NADPH phagosome oxidase, they convert it into reactive oxygen species. Now superoxide dismutase convert it into H2O2, and ultimately these particular ROS is very harmful for the pathogens. Okay, for the pathogens this ROS is very harmful. Not only ROS, but there is also reactive nitrogen species or RNase. These reactive nitrogen species are generated by L-arginine. It turns out macrophages had a lot of L-arginine transporters. So they take up L-arginine and they convert it into nitric oxide, and ultimately it gets converted to peroxynitrile. So these reactive nitrogen species also kill the bacteria. So this is known as oxidative burst by macrophages. So this tells you that after engulfing, how the pathogen is killed. I mean, the simplest machinery was the lysosome, but there many pathogens have devised way to evade the lysosome. So this is a fail-safe way by which they can sort of fight the pathogens, or they can kill the pathogens. So everybody understood this part? I hope so. So once we had learned about macrophages, not totally because we haven't looked at the toll-like receptor yet, but let us talk about the dendritic cell in a little bit, and then we'll go back to the toll-like receptors. Okay? Yes, ROS is actually harmful for our body, but when it is generated in a prolonged fashion and when it is persistent for a long time, in case of macrophages, the ROS production is temporally restricted. That means it is, it's like a burst, it's like a pulse. So ROS is generated in a pulse and it does not persist for a long time. Eventually, inside the macrophages, there are ways by which they can counter ROS. So they just need to make the ROS to kill the pathogen, and it is later on neutralized. Okay? So this is very important question what Sommaya is asking. How TLR recognize PAMs and what happens? Is there any conformational change? What are actually happening? Obviously, your answer can be done by crystallizing a PAMP with a corresponding TLR. But before that, we need to know that what kind of TLR are able to recognize what kind of PAMs. And for that, you have to wait a bit because we need to finish a little bit about dendritic cells and then we move on. So this dendritic cell also have toll-like receptor just like the macrophages. Now, dendritic cell can recognize specific patterns, and we have looked at these patterns. Some of them are bacterial flagella, some of them could be like lipopolysaccharides, teichoic acids, some of them could be, let's say, DNA or RNA material. So all of these things can be patterns or pathogen associated patterns. Also there are certain receptors which can determine damage associated molecular patterns, or known as DAMPs. Okay? Okay? Yep. So if a person has infection and if the macrophages are active, so the ROS would be locally increasing, not everywhere in the body, but only inside the macrophages, only in a localized tissue. For example, why do you feel inflamed when a mosquito bites, or let's say an insect bites in your hand? There is redness, right? So that's a classical signature of inflammation. So inflammation is also an innate immune response. It's good for your body, but some of these manifestations we think it's bad. The swelling, the redness, the itch, all of these things are actually a manifestation of the inflammation, that already your innate system has started encountering these pathogens. So that's the signature. Okay, so you guys understand a little bit of that? Okay, so let us talk about toll-like receptors. Okay, so let us try to understand that how toll-like receptors work. Okay guys, so these toll-like receptors, let us take an example that we have taken right now. So this toll-like receptor is actually, let's say, TLR4. I haven't talked yet about the discovery story, I'll tell you. So eventually these toll-like receptor would be recognizing bacterial flagella, a portion of the bacterial flagella, right guys? Yeah, so this is basically the ligand for these receptors. So though it is depicted that the entire flagella is detected, it's not the entire flagella. Some portions of the flagella is detected by the TLR. Okay, that's the simple way. Now this TLR has 3 domains. So first of all, you see here, this is the transmembrane domain. Okay, so this is basically transmembrane domain. This curved like structure is actually known as leucine rich repeats. So they have a hell lot of leucine in their particular in this fold, and they one type of common interaction is hydrophobic interaction, and they actually recognize lipids. That makes a hell lot of sense, right? Okay, now here you have the TIR domain. So this domain is known as TIR domain. So this is known as toll interleukin 1 domain. So it's a characteristic domain. We would learn about it more when we talk about interleukins under the big topic cytokines. But today we are not going to talk about it in very much detail. So this is the toll-like receptor. Now the toll-like receptor would bind to some kind of adapter, and this adapter has something in very much common, that is the TIR domain, and they interact with each other via these TIR domain. Okay, so this particular molecule is known as MyD88 or myeloid differentiation factor 88. Okay, there are fantastic stories about all of these, but we are not talking about this in details. Okay, now this myeloid differentiation factor has 2 domains. So one domain is known as death domain, this one, and this domain is basically the same TIR domain. So you can understand the signaling begins when there is an interaction between TIR and TIR domain, right? Now these things, actually this death domain can then activate several molecules. Okay, in this case these molecules are nothing but some kinases. Okay, so these kinases are known as IRACs. Okay, and TRAF. So ultimately what happens, guys, these kinases activates further downstream molecules such as TAC and TAB complexes. These are absolutely unnecessary informations that I am giving it to you. But ultimate what is important is the activation of the transcription factor NF kappa beta. Okay, so this NF kappa beta is a transcription factor. So this is highlighted and this is important. Okay guys, yep, so this is super important. So the NF kappa beta, what it does, NF kappa beta obviously binds to DNA and give rise to gene transcription. Transcription of what? Transcribe pro-inflammatory cytokines such as interleukin 1, interleukin 6, TNF alpha, etc. So all of these things are actually generated by NF kappa beta signaling, right? Now guys, this is one of the way. So we have looked at one way, right? So don't think this is only the signaling way that happens inside the cells. There are multiple different ways. So let us take an example of different kind of PAMP. In this case, the PAMP is present in an endosome, in an endosomal vesicle, okay? In an endosomal vesicle. This is an endosome, okay? Yeah. This PAMP is in the endosome. Now, in the endosome, the viral dsRNA, let's say this is a RNA virus or dsRNA virus. So, viral dsRNA would be actually detected by specific TLRs. Okay. So, dsRNA would be detected by TLR3. Okay. Now, this viral particular detecting TLR would activate MAP kinase signaling and ultimately lead to production of AP1 family gene that helps in the proliferation process of the macrophages. Also, they can activate in a sequential fashion the interferon response factor 1, IRF1 or IRF7, etc. Okay, so these interferon response factors actually lead to production of interferons. So I hope we would talk about interferons eventually, guys. Okay, so everybody is still with me? Everybody is still with me? It's not bored yet? So now what we would do, we would solve some question. Okay, guys? So ready for that? So this is the structure of the toll-like receptor. Before that, I wanted to convey something. This is the structure of the toll-like receptor. So they pretty much look like a question mark, but this is how their crystal structure look like. And in red, what you can see is some kind of bacterial flagella portion is embedded in that. So they could either be homodimers or they could be heterodimers. Okay. For example, TLR4, TLR3, these are homodimers, whereas TLR1 and 2, which recognize lipopolysaccharides, they are actually heterodimer. Okay. So let us try to take some example of these TLRs. So most of them are homodimer. Among them, one very important TLR is actually TLR4, because TLR4 turns out to be present inside the endosomes. Also, they are present in a surface, right? So TLR4 is very important and unusual, and they recognize flagella, right? Flagella protein, flagellines. Okay, and they form homodimers, right, guys? So now let me give you an example of a TLR which forms heterodimers, such as TLR1 and TLR2. Okay, guys, TLR1 and TLR2, and they recognize lipopolysaccharides. Okay, lipopolysaccharides. Now there are many others, like there are quite a lot of them. So let me give you some more examples. For example, these lipopolysaccharides could be also derived from several parasites or anything, so they detect all of that. So for example, TLR2 and 6. Sorry. Yeah. So for example, TLR2 and 6, they recognize fungus or fungal proteins. So don't think our enemy is only bacteria or viruses. Our enemies are actually also funguses, parasites, etc. Right? So this is very important. So then we talk about another TLR. So you guys are with me? Okay, so already Soumya has asked that how these kind of different TLRs are produced. Actually, there are 10 TLRs, and these TLRs are encoded by several genes. Now the question is, all of these genes are kind of expressed. So all of these combination of receptors are present on the surface, okay? On the surface, okay? Now I'll come to a very interesting story about TLR. I'll tell you guys. So now guys, there is also, let's say, TLR5. This detects bacteria. Okay. So also TLR1 and 3, they also detect bacteria. Sorry, TLR1 and 11. TLR1 and 11, they also determine bacteria. So we pretty much understand that these TLRs can determine many things, right? Also viral dsRNA that are detected by, I already said, TLR3, whereas TLR8 they recognize viral ssDNA. So viral DNA too. Obviously they are patrolling police officer, and they need a hell lot of energy for themselves to go towards other locations, such as an army base camp, which is the lymph node, right? Inside the lymph node you have T cells, B cells, etc. So they would now go towards that, right? So now they would migrate all along the bloodstream and go towards the lymph node, right guys? So inside the lymph node they would do many things. They would activate actually T cells would activate B cells, eventually plasma cells would be produced. So these events happens inside the lymph node. So you clearly understand that this is the bridge between the innate and adaptive immune system. Like we said, these innate and adaptive immune systems are like the kind of a front line and the backup line. But if there is no communication between the front line and backup line, how does the backup line would understand that and there is an invasion, because backup line is not in that war zone, right? They need to understand. So they need a messenger, and these cell types are the messenger, right? So the dendritic cells are mostly motile. They are pretty different from macrophages because they capture antigens from one place, one tissue space, and they totally take it to the lymph node, which is the army base camp, for the recognition part. So this is something unique about dendritic cell, but not true for the macrophages. Apart from these things, what they would really do, but before that I'll tell you that these macrophages were like police officers. They go to these army base camp, they interacted with the armies, and they let them know that there is something happened, some kind of invasion has happened. The situation might be in control, or they should be recruited. But if they don't know what type of pathogens are invaded the situation, then they had no idea that how actually these army rangers can fight them. So by providing those molecular signature or information about those molecular signature, they're priming these army rangers or giving them intels about the attack, such that they can really frame their responses, right guys? So we pretty much understand this part. Now another unique property about dendritic cells, since we are talking, is the cross presentation. Generally we looked at dendritic cell from a point of view of classical antigen presenting cell, or APC, right? Professional APC. That means they simply display their pathogen derived antigen on MHC2 molecules. Eventually it is already shown that they can also express class 1 MHC. They generally report to the T helper cell, not the T cytotoxic cell. But some special cases, they can also interact with cytotoxic T cells, the T killer cells. Generally they don't do so. And cytotoxic T cells generally are trained to recognize MHC class 1. Now they should ideally present MHC class 2, but sometimes dendritic cell also present it on class 1, and this is known as cross presentation, because generally they are trained for MHC class 2, right? That is why the crisscross thing is happening. But the thing is, there are certain problems when we talk about more about dendritic cell, we would understand what is the problem of cross presentation. Today I wanted to give you a flavor or a little bit foundation that they are able to do cross presentation. What are the consequences of cross presentation? Is it advantageous? Is it disadvantageous? All of these things would be clear as we go along. But it's only the second class, so we have a lot of time, right? But the benefit is they can really quickly, let's say there is a virus infected cell, they can capture those pathogens in the tissue space and quickly go there into the lymph node without wasting time, and they can show it to the cytotoxic T cell such that the response is faster, the cell mediated response is faster. They ensure that. So that is one way, but there are potential disadvantages, and that's why all the dendritic cells don't have this ability. For example, you're a student, I'm a student, we might have a car, but we all might not have a license to drive the car, right? So somebody really need to give us a license such that we can drive the car. Similarly, presenting antigens on class 1 MHC requires permission, license, and this licensing has to be done, and how it is done, we'll talk about it later. So guys, everybody is with me, because right now we are going to start some questions, okay? We are going to start some questions. So try to answer these questions. Which of the B cell is actually derived from lymphoid progenitors? Not the myeloid progenitors. So everybody understood this point. So if you don't understand or forgot it, then what you should do, you should go back to last day's lecture. Okay, the development and differentiation of macrophage takes place respectively in bone marrow and thymus, bone marrow and tissue space, thymus and lymph node, thymus and spleen. So you have to choose the correct option. So Hiten Navani is saying it's A. So anybody else is saying something else, or everybody thinks that it would be A? Pritam Mitra is saying A, Anubhav is saying A, so everybody is saying A. Is there any other options than A that people think? Shipra Singh is saying B. So is anybody saying B? So let me give you the answer. Correct answer is B. So they develop in the bone marrow and they mature or differentiate into the tissue space. They are not T cells, so thymus यहाँ पे नहीं लाने का. Okay, so you can't consider them to be the thymus. Thymus is the developmental school for T cells, not macrophages. Macrophage differentiates into the tissue space. We already talked about it, right? Monocyte to macrophages, right? B is the correct option. Which of the following is not an antigen presenting cell, like professional antigen presenting cell? Macrophage, dendritic cell, B cell and mast cell. So these are very popular type of question that can be asked in any exams. So tell me guys. So D is the option. Great guys, D is the option. So B cell, dendritic cell, macrophages, all of them have class 2 MHC. But this one does not have the class 2 MHC expression, so they are not antigen presenting cells. So this question came in December 2006 in CSIR NET exam. So let us take a look quickly. Which cell is involved in antigen clearance non-specifically by engulfing them? Tell me guys, what could be the cell type? So they are saying which are the cell types which is killing in a non-specific manner just by engulfing them. So you just need to understand that they are engulfing, okay? So tell me the answer. So Srinath is saying it would be D. Many of the people are saying D. Actually the answer is D. So they are phagocytotic cells such as dendritic cells. Macrophages, right? They are not cytotoxic T cells. Cytotoxic T cells are very specific. They can only recognize antigens from class 1 MHC. Okay, they don't non-specifically hit anybody. Okay, otherwise there would be autoimmune response. Okay, sorry about this bad snipping quality, but anyway, name the first cell which are recruited at the place of infection. Which one would be the first cells? So D is the answer, macrophages, right? So macrophages are like patrolling police officers, so they engage first. But now I just change the question a little bit and saying that tell me the order, right? So you write in the comment that A, B, C, D. If you think that A comes first, then B, then C, then D, then write A, B, C, D, or let's say write D, C, B, A, something like that. So try to give me the order. So Saloni is saying D, A, C, B. So let me tell you, the order is actually D, then C, then A, then B. Okay, so first macrophages are recruited. Macrophages itself tell the neutrophils to come. They said, okay, come. And then neutrophils come to the region. They are also phagocytotic. We'll talk about neutrophils in a moment. Then NK cells, they are also part of these innate immune response. And lastly, basophil might be recruited, might not be recruited, so they are last. In special cases, they are recruited. Okay, so everybody understood this concept. This is important. Which of the following cell type possesses polymorphonucleus? This is super easy. So tell me the answer, guys. So I want everybody to attend this question and try to answer. Be it wrong, be it right. Come on, answer it. So everybody would understand this is neutrophil, right? They have polymorphonucleuses. Okay, mast cell has receptor for So are you giving the answer for this question or previous question? Mast cell has a receptor for IgE, A, G or M. IgE, right? So this is very important. I'll tell you about mast cell later on when we talk about allergic responses, but today let's focus on something else. Histamine secretion by degranulation from which cell leads to immediate hypersensitivity? Which of these cell types? So see, these are the same questions. It is also mast cell that degranulates after it binds to IgG antibody via Fc receptor. So mast cell is an important concept, already you know, right? So mast cell, eosinophil, neutrophil, basophil, all of them take part to some extent in allergic responses and inflammatory processes. As we go along in this course, we would understand all of that. So today let's get the overview, because we are only 2 classes, right? We have only 2 classes in this topic. So gradually we would understand it better. But if you stick to it, after 1 month you would be an expert in immunology. You would understand A to Z of the immunology, trust me. So these cells, whenever they are in front of you, you have to think that they mediate inflammatory response. They also modulate adaptive immune responses, right? So innate immune response and adaptive immune response are different. So tomorrow obviously the class would be adaptive immune response. Now they are different, but they are interconnected. One modulates the other. So today we would look at a little bit that how adaptive immune system would start responding, and then we would take it from there tomorrow, okay? And they are involved in allergic responses, already I told you. So they can tell you this kind of question: which of these cell types are involved in allergic responses or not involved in allergic responses? Indeed, same question was asked in IIT JAM or JNU several times. Okay, let's talk about some other questions. For example, this one: which one of the following is not a true statement about the macrophages? Okay, so read these options and tell me which one of these are not true for macrophages. You have to identify the false statement. So some people are saying B, some people are saying C. Lock किया जाए क्या? So B is the correct answer. They are not differentiated from basophils. So they are not differentiated from basophils. They are differentiated from monocytes, right? So B is the correct answer here. Moving on to the next question. So which statement about dendritic cell is false? So read this question and tell me. So these are some questions that I made for you guys for your better understanding. So this kind of involves your basic understanding how you can do it. Okay. Yeah. So C is the correct answer here, right? Because indeed dendritic cells are professional antigen presenters. So with a collection of all these kind of most important questions, I have an immunology booklet, and this booklet is available only at the rate 100 rupees. So if you want to take, just email me or write in the comment that I need a booklet, or better it's like you send me an email. This would really help your or boost your understanding, because see, many of you guys are not answering properly. That means there is some kind of problems with your understanding. So in order to test, you need a booklet. Which of the following statement about the basophil is correct? Basophil are involved in development of amplification of type 2 responses against helminth. Basophil is, Basophil interacts with T cell and activates them. So you need to find the correct answer. Okay, basophils are derived from lymphoid lineages. Basophils are involved in type 3 hypersensitive response. So guys, could you understand the correct statement? Like, which one is the correct statement? So for this question, right? So the correct statement here is actually this one. They are not derived from lymphoid lineage, they are from myeloid lineage. This is false. They don't interact with T cells. Who interact with T cells? Macrophages, dendritic cell, not they. Okay, basophils are involved in type 3 hypersensitive response. Okay, you guys don't know this answer, but this is also false. We'll talk about it. So everybody, do you guys understand these concepts? Now shall we move on to a little bit towards neutrophil? That's the next event or the chronology in which these cells would be recruited. Now neutrophils are circulating inside the bloodstream. They have to come out in the place. Okay, so already they are same, let's say they are like detectives who play an important role. So the patrolling police officer would take over when there is a fight, but the detectives can eventually join them and fight with them, right? So these are the neutrophils. They are rolling all across the blood vessels, and there are RBCs as well. Now they have glycoprotein receptors which can bind to specific receptors on the blood vessels as well. The capillary endothelial cell also have adhesion molecules, and these adhesion based interaction help them to roll. Okay, now if these selectin or S-legs, these kind of interaction are too much in nature, then what would happen? They would slow down. So these are like a brake, right? You increase the friction of these rolling. There are many more interactions such as LFA1, ICAM1. So these adhesion based interaction prevent these neutrophils to roll. Okay, they prevent them to roll. They tell them to now slow down, stop here. Then there would be certain signaling via these receptors as well, which lead to a cytoskeleton rearrangement. Now let us try to understand what exactly happens in format of an animation. So they kind of try to move out, they kind of squeeze their way out, right? They squeeze their way out of these blood vessels. Already look at the blood vessels. The blood vessels are pretty like scattered, like they are like dilated. The spaces in between them are now more, right? So this happens due to vasodilation. So many inflammatory cytokines secreted by macrophages lead to these kind of changes, and they actually bring more of these cells in the position, right? And this vasodilation is triggered by these inflammatory cytokines. So these are inflammatory effects, okay? And they now follow a chemokine gradient and sort of move towards that. And this is secreted by macrophages. So macrophage basically signal people, okay? So now macrophage, dendritic cell, neutrophils all are near the site of infection. So macrophage, dendritic cell are phagocytotic. Neutrophil are also phagocytotic. They would come here and they would kind of engulf the pathogen and take it in. And they have kind of similar mechanisms like macrophages. Lysosomal degradation would happen for these bacteria, okay? But they don't have toll-like receptors. They cannot like display it, or they cannot work like a bridge between the innate and adaptive system. So you can understand or appreciate their functions, right? But neutrophils has specific responses, especially for fungal responses, parasite responses. Neutrophils are very important. So tomorrow we'll practice some questions on these part. Next, guys, there is natural killer cell. Natural killer cells are the lethal weapon of this innate immune system. Lethal weapons means they are lethal weapon. They are born to kill. They are army rangers who would shoot to kill anybody. They won't discriminate that much. Only you really show them that, okay, we are friendlies, then they won't kill. Otherwise, they are ready to kill. Their default state is a killer. Okay, so they are generally, let me remind you that they are developed from the lymphoid progenitor, just like the B cells and T cells, right? So even if we said that lymphoid progenitors broadly give rise to adaptive immune component, but NK cells are derived from lymphoid progenitor, which can give rise to an innate component. So it cannot be generalized, okay? So this is important. So they are assassins. True. Now these NK cells, they have barrage of receptors. They have inhibitory receptors, they have activatory receptors. Most important, they have their signature. So by looking at NK 1.1, you would understand this is an NK cell. They also have Fas ligand, which is helping in the apoptosis. So what happens when they really engage? They kill the cell. Okay, they kill the cell. So these activatory and inactivatory receptors are very important. Activatory receptor would always tell them, okay, kill. Inhibitory receptor would say, okay, don't kill. Okay. So, but how the NK cell knows that something wrong? A cell is infected with virus. So whenever the cell is infected by the virus, generally what happens, these viral peptides are displaced on class 1 MHC molecules on the cell surface. That is recognized by whom? CD8 T cells. Now this is adaptive immune system. But sometimes adaptive immune system also falls. They fail to recognize these because some bacteria devise strategy where they can down regulate these particular class 1 MHC. So for a CD8 positive T helper cytotoxic T cell, everything seems to be all right because no viral pathogen or viral antigen is displayed. Everything seems to be right. He thinks everything is right. But NK cell would think, no, there is something wrong. I should now engage. So any cell that cannot display any antigen, self antigen onto its surface, they would be killed by NK cell. Okay. So that is how NK cell work like a fail-safe mechanism in the killing response. So NK cell engage in that case. So generally these MHC expression would tell them to disengage and don't kill, but whenever the MHC1 is not there, their default state is killing because they are all assassins, right? Okay, they would secrete perforins and granzymes, which would in a cascade of pathway activate caspases. So they can either activate cytochrome C and then lead to caspase activation. They can also activate initiator caspase such as caspase 8, which ultimately lead to caspase 3 activation, which is the executor caspase. About apoptosis and necrosis I haven't talked yet, but we'll talk soon. Now NK cells also secrete cytotoxic granule, okay? This would kill the cells. So NK cells, they have activatory receptor and inhibitory receptor, just to summarize. So both kind of signal is there. So it's like whenever you have both, you have a brake but have an accelerator. Generally you accelerate, but when you have a brake, the net result is 0. Right? You have your brakes removed, you accelerate because your default state is acceleration, right? That is how you engage. So before we understand more about that, so whenever there is a MHC down regulation in cultured cell, in a virus infected cell, you down regulate MHC, there was apoptosis triggered by these NK cells. But in this isolated culture, when you restored the MHC, Okay, there was no death, almost no apoptosis. That means, see, it is not a body, so already the CTLs won't be here, the cytotoxic T cells won't be here, only NK cells would be present in this culture, right? So now they have a break, and that's why there is no death. Exactly. So there are a barrage of activatory receptors and inhibitory receptors, and especially for tumor cell, against tumor cell, NK cell therapy is very popular these days, and they are really helpful. Okay, NK cells are very important. Now let us understand a little bit more about other things, complement proteins. So complement proteins can also decorate any pathogen and opsonize them. So this is the term, opsonization. Later on, with the coordination of the humoral immune response and, let's say, complement system, the bacteria can be burst. There could be osmolarity disbalance and the bacteria could burst, and that's how they can encounter pathogen. Okay, and obviously this is a link between humoral immune response and the innate immune response. The complement proteins are innate immune response proteins. I'll tell you something about interferons, not the story today, but tomorrow maybe the story. Interferons derive their name from their interfering capabilities. Interferons are now widely used for cancer, osteoporosis, rheumatoid arthritis, and hepatitis C treatment. Okay, so interferon has a huge clinical potential. So what are interferons? Where do they come from? How does interferon work and why they are beneficial? So interferons are generally secreted by the neighboring cell in response to a viral infection. So let's say this particular cell on the left hand side is infected by a virus. So this cell would generate an alarm signal, that is basically the interferon, such that the cell in the near vicinity would know that there is a viral infection, and they can prepare accordingly and can be immune against the virus. So what type of cells really interferon make? So if you compare before that, if you compare immune system to be a defense system, so you can clearly understand they need to communicate. So interferon is one of the way of communication between cells and cells, right? So there are cell-cell communication methods. Now the interferon is actually one of the family member of the 6 cytokine families, and even interferon has subdivisions which we don't need to understand right now. But interferons are secreted from the cells who is going to be dead and who was infected by the cells, so the cell cannot be surviving anymore, so it saves the nearby cell by secreting the interferons, and the interferon signaling leads to antiviral modules or antiviral mechanisms which can really fight against the viruses and prevent the viral entry in the cell, or even if the virus enters, it makes sure it cannot replicate or increase their number, right? So this is how interferon works. So I hope you enjoyed this whole lecture. If you like this lecture, give it a quick thumbs up, and don't forget to like, share and subscribe. Please share it with your friends. It's absolutely free. I hope this lecture was useful. Okay guys, and do let me know in the comment. If you want to join a WhatsApp group, do give your number in the comment section. I can add you. Okay guys, bye. Or you can email me your number.